Good morning to all and welcome to Ratnam Group of Institution. I am Mrs. B. Mohana Priya from the Department of Biotechnology. I am handling the subject pharmaceutical biotechnology, and now today we are going to discuss about the topic microarray technique. So this is a fascinating topic and numerous names are available for this technique. Example, biochip, organ chip, DNA chip, numerous names are there. So all will be coming under one category, that is microarray technique. So what is the main outcome of this topic will be, we will be knowing about what is meant by DNA microarray technique and some of its application related to pharmaceutical industry. So coming to main components of this topic is about introduction part and types of microarray, mainly DNA microarray, what is the main principle behind in this DNA microarray technique, and some of the steps involved to perform this microarray technique, and types of cDNA. That is, cDNA is nothing but complementary DNA chips. So it is called as cDNA chips, and then oligonucleotide chips is also another type of DNA microarray technique, and applications, advantages and disadvantages, and single nucleotide polymorphism technique also we are going to discuss today. So coming to introduction part. So what is meant by this DNA microarray chip is, it will be very compact chip. It is at the size of the chip will be about nearly a microscope slide will be. It is the actual size of this chip. And what this chip will be containing is that it will be containing a numerous and thousands of DNA samples will be present. That is, it is present in which fashion is that grid manner. So it will be present in the grid manner, present on a small, complicated and very chip. And then what is the main principle behind in this technique is the hybridization. Hybridization is the main process involved in this technique. And what are all the other names as we discussed above? So biochip, DNA chip, organ chip, these are all the synonyms of this category. And then history, from where this technique has been came is that nothing other than that Southern blotting. Southern blotting is the main technique where this DNA microarray chip technique has been came. So what is the thing involved in this technique is that additionally the DNA fragments will be attached and then it will be probed with the known DNA sequence. So known DNA sequence will be taken from the patient sample. So it will be different part we are discussing in the future classes, and the main miniature of this DNA chip technique has been came from at the year of 1995, and then entire genome profiling has been done for a microorganism Saccharomyces cerevisiae, and it has been published at the year of 1997. So coming to the types of DNA microarray. So numerous types are there, example like protein, antibody, tissue and chemical compound basis. So main thing is the hybridization. Hybridization of the nucleotide will be plays an important role in this type of technique. So coming to another thing is that how this will be performed, how this type of diagnostic procedures are performed is that we will be taking a DNA sample from a patient who are suffering from, example, disease like cancer, and then the sample can be taken from the sample, and then procedure is that we will be converted the DNA sample in single stranded molecule. The main thing is we should convert it into the single stranded DNA strands, and then we will be adding to the DNA chip. So the DNA chip will be ready with the another type of dyes will be there. So fluorescent dye will be added to this chip. So what is the reason behind this addition of this DNA sample with the dye is that the coloring will be helpful to identify whether the sample is containing any cancerous substance or any other disease or not. So this is the main thing. So coming to the another part is that how we are doing this is the how technique is that the unknown DNA will be inserted into the chip and it will be allowed to hybridization to the known DNA sequence. So known sequence will be present in this DNA chip and unknown will be taken from the patient sample. After that the color formation will be formed due to the presence of the fluorescent dye. So the target DNA molecule which will be only binded to the complementary, that is cDNA will be binded. So we can able to find out with the color change. So what are all the colors means mainly about green, yellow, these are all the main colors. It will be helpful to find out whether the patient is affected by the disease or not. So coming to the another thing, designing of this DNA microarray. So what are all the steps and what are all the designing of this microarray technique is that it starts from the collection of the sample. So we are going to collect the sample from the patient who are suffering from different type of disease, and then we are going to find out diagnostic purposes of this using this DNA chip. So next to the collection of the sample, we will be going for the purification. The sample will be loaded with different types of biocomponents. So we need to separate the DNA, RNA, mRNA sample with the help of the purification steps, and next is the reverse transcription. With the help of the PCR, that is polymerase chain reaction, we will be performing the reverse transcription process. After the process only, we will be going for the coupling. During this coupling process only, we will be adding the fluorescent color, that is colored dyes will be adding to the sample, which will be helpful to find out the results. That is with the help of the intensity ratio, we can able to find out the results. And then hybridization. So after this coupling of this sample with the particular dyes, the coupling reactions will be takes place. So after with the usage of the scanner, so laser scanner will be nowadays we are using with the help of the scanning. Then we will be going for the intensity ratio calculation with the help of the systematic analysis. So in this technique, we are utilizing presence of the sample from the patient, then to the purification step, then RT, that is a reverse transcription process with the help of the PCR technique, hybridization and washing of the extra samples will be present in the chip, and then to the scanning also will be done, then intensity calculation with the help of the systematic analysis. So now sample from different types of patients we are going to collect. So if we are suffering from any type of disease, means we can able to find out the help of this DNA chip. So this technique will be very closer to find out whether the patient is suffering from numerous type of disease or not. And then purification with the help of the numerous purification process will be done to separate the protein separately, DNA particles and then mRNA particles. And then in this step only we will be able to find out the single stranded. So from the double stranded molecule, we want to able to calculate the single stranded molecules. So it is an important category, because to bind to the chip we need only single stranded molecule, that is a complementary DNA sequence should be used. So and then reverse transcription with the help of the PCR and labeling with the fluorescent dye will be then, and then later hybridization, scanning and normalization and analysis of the results. So types of DNA. So what are all the main two types is the cDNA. cDNA is nothing but complementary DNA microarray and oligonucleotide based DNA array. So in the case of cDNA, what is meant by the cDNA based chips means in this type what will be we are utilizing is that cDNA, that is complementary DNA sequence we are utilizing in this chip. So it is called as cDNA or probe DNA. So main thing is that amplification will be done in the case of then cDNA chips with the help of then PCR analysis, and then the immobilized one will be made onto the support, that is solid supports. This DNA chip will be either it will be made up of any silicon or glass material. So the main thing made up of this material is that either glass or silicon material. So the probe DNA loaded with the help of the spotting. The spotting can be done with the help of any material like robotics. So because the sample will be collected from patients suffering from different type of disease, so we will be very precautious to handle this type of samples. This spot will be taken and manually it will be loaded to the DNA chips. So when the DNA spotting will be done means it will be extra things will be washed up and we will be going for the second cycle. So numerous cycle will be takes place in this case of a DNA chip technique. So this picture clearly shows the comparative microarray hybridization starting from the collection of the sample and extraction of mRNA from the sample, and then reverse transcription, the RT-PCR we are doing the reverse transcription process, and fluorescent labeling of the DNA sample will be done, and then hybridization process. So how the DNA sequence will be hybridization, the chip will be takes place, and then scanning. So scanning also plays an important role. So because with the help of the detector, so the scanning will be done, the intensity ratio will be calculated easily. So coming to the advantages and disadvantages. So what are all the application and advantage disadvantages means application. So in which field we are mainly applying this DNA chip technique is that it will provide thousands of genes from a single experiment. So it is a single experiment. So we can also consolidate this experiment with only one tape. So it will be very fast and easy to find out the results, and huge steps will be done because very closer to the disease can be easily calculated with the help of this DNA chip technique. And different parts of DNA can be also used for this study of gene expression. It is also play an important advantage. So different parts of DNA can be utilized in this technique. Coming to the disadvantages, some of the disadvantages of the technique is that, so the main disadvantages coming, the shelf life time of this DNA chip is very low. So once we are preparing this DNA chip is that we should perform the experiment at the same time. We can't able to withhold for long time. So shelf life time is a very main disadvantages of this technique, and then it will be more expensive. So expensive, the cost effectiveness will be very high. So it is also one of the disadvantages of this DNA chip technique. And then the production of too many results at a single time also it will require a huge time. So it will also take some time, because we are inserting numerous number of genes or numerous number of DNA probes into the particular DNA chip. So the hybridization process will be takes place between the numerous sequences. So the production of the too many results will be at a time it will require, it will take some more time. So coming to the application, in what all the fields we are applying this DNA chip is that mainly in the toxicological research and drug discovery, mainly disease diagnosis. So disease diagnosis plays an important role. Either the patient is suffering from a particular disease or not, we can able to find with the help of this DNA chip technique. And then comparative studies. So once we are going for analysis of genome studies, means comparative genome analysis can be done with the help of this chip microarray technique. And microarray technique is also helpful in gene expression studies. Mainly gene expression level of a particular organism can be done with the help of this DNA microarray technique. So these are all about the introduction, some of the steps involved to perform this DNA microarray technique, advantages, disadvantages and the application of this technique in the field of pharmaceutical industry. Thank you.